At dusk, a silver eel leaves its river and enters an ocean without roads. Destination lies far offshore, within the rotating waters of the Sargasso Sea. No adult guide leads the departure, and no visible landmark spans the Atlantic. Yet, mature eels from distant coasts begin converging toward the same broad region. The mystery is not whether they migrate, but how they hold a useful course. European eels may travel thousands of kilometers before reproduction ends their known life cycle. American eels make a shorter crossing, but face the same featureless darkness and moving water. If you followed one beyond the continental shelf, its trail would soon disappear. Archival tags sometimes reveal depth, temperature, and light, but rarely an uninterrupted route. Each recovered record becomes a broken sentence from a journey mostly hidden below waves. The travelers themselves change before departure, enlarging eyes and preparing muscles for endurance. They stop behaving like river residents and become animals organized around a single passage. Night after night, their headings must resist currents capable of displacing them hundreds of kilometers. Navigation, therefore, begins as a moving problem inside another moving system. The Sargasso Sea waits ahead, bounded not by land, but by great Atlantic currents. Freshwater eels belong to the genus Anguilla, although much of their lives unfolds at sea. Anguilla grows across Europe and nearby regions before beginning its westward reproductive migration. Anguilla rostrata develops across eastern North America and the Caribbean before moving toward overlapping spawning waters. Their transparent, leaf-shaped larvae are called leptocephali and drift away from the Sargasso region. Ocean currents carry these larvae toward continents over many months, transforming distance into development. Near the continental margin, they become glass eels and enter estuaries, rivers, or coastal habitats. Years of feeding follow before hormonal and anatomical changes prepare the return migration. Skin darkens above and brightens below, improving camouflage in the open ocean. Eyes enlarge, the gut regresses, and stored energy becomes the fuel for travel and reproduction. These changes are directly documented, but they do not reveal the eel's internal compass. Researchers can confirm departure timing by traps, fisheries records, and electronic tags. Many silver eels leave during dark, wet periods when river discharge helps carry them seaward. Once offshore, tagged individuals often alternate between deeper days and shallower nights. This daily vertical movement may balance predator avoidance, temperature, buoyancy, and energetic costs. The pattern also exposes eels to changing light, pressure, currents, and magnetic conditions. Any of those variables could provide information, yet exposure alone does not prove navigation. Mainstream interpretation favors several cues working together rather than one perfect biological instrument. Earth's magnetic field is a leading candidate because it varies predictably across broad geographic space. Laboratory experiments show that young eels can alter orientation under experimentally shifted magnetic fields. Those results support magnetic sensitivity, while leaving the adult oceanic mechanism incompletely tested. Smell may help near rivers and coasts, where chemical gradients remain stronger and more distinctive. Far offshore, odor cannot easily explain a basin-scale course toward a shifting, spawning region. Celestial cues are possible near the surface, but deep nighttime travel limits their continuous use. The strongest picture is layered navigation, with different cues becoming useful at different scales. To test that picture, scientists first compare actual headings with winds and ocean currents. If eels simply drifted, modeled tracks would follow water movement more closely than many records suggest. Some tagged eels maintain directions inconsistent with passive transport, indicating active swimming and correction. However, tag records are sparse, and release sites can alter an animal's initial behavior. Pop-up tags detach at programmed times and transmit summaries after reaching the surface. They can reveal broad movements, but location estimates may remain too coarse for subtle turns. Few devices complete the full migration, especially across the longest European eel route. You can reconstruct a corridor from fragments without seeing how each decision was made. Currents may assist progress on some nights and oppose it on others. An eel could combine an inherited directional program with local corrections from magnetic or hydrodynamic cues. This hypothesis fits the scale of the journey, but direct proof requires controlled adult experiments. Magnetic maps also change gradually, and storms do not erase them as they erase surface signs. Yet magnetic sensitivity need not mean that eels perceive a detailed map like humans do. They may detect inclination, intensity, or polarity as simple instructions to turn or continue. Experiments on juvenile stages suggest orientation can shift with simulated geographic magnetic conditions. Whether mature silver eels use identical rules during reproduction remains an open question. Water temperature offers another boundary, because fronts and current systems outline changing Atlantic habitats. Temperature alone cannot identify the Sargasso Sea, but it may signal unsuitable or favorable water masses. Current shear could also provide direction through turbulence sensed by the body or inner ear. That possibility remains difficult to separate from ordinary swimming responses inside laboratory tanks. Moonlight and polarized light may contribute near the surface, though evidence for adults is limited. No single cue currently explains departure, offshore bearing, correction, and final spawning site recognition. The scientific challenge is therefore to test combinations without turning plausibility into certainty. Each missing tag narrows the data, but it does not transform an unknown path into failure. Each successful record shows movement, not necessarily the sensory calculation beneath that movement. The journey matters because navigation connects inland food webs with a distant oceanic nursery. Eels accumulate energy in rivers, lakes, marshes, and estuaries before exporting it into reproduction. Their larvae then return marine production toward continents through the circulating North Atlantic. This exchange depends on adults reaching suitable spawning waters at the correct season. Dams, turbines, pumps, and fragmented rivers can prevent migrants from reaching the ocean at all. Coastal pollution and parasites may further reduce the condition needed for a long-fasting swim. Climate-driven changes in currents could alter larval transport, even if adult navigation remains effective. The Gulf Stream and associated circulation are not conveyor belts with fixed speed or position. Small shifts in timing can change where larvae arrive and whether coastal habitats receive them. Navigation research, therefore, intersects with conservation long before an eel reaches deep water. If you protect only the destination, blocked rivers can still empty the migration at its source. If only rivers are repaired, changing ocean conditions may still weaken the returning generation. European eel recruitment has declined severely, and multiple pressures probably contribute across its range. American eel trends vary regionally, reminding researchers that related species face different human landscapes. Management benefits from knowing when silver eels migrate and which offshore corridors they use. Those corridors could reveal periods when fishing, turbines, or construction create concentrated risks. They could also show whether migrants from different rivers merge early or approach separately. Population mixing in the Sargasso region is supported genetically, but individual routes remain poorly resolved. This makes every complete migration record unusually valuable, especially when paired with environmental measurements. The eel is not solving an abstract puzzle. It is completing the only reproductive journey available. Future evidence must follow adults farther without burdening their streamlined bodies or changing their behavior. Smaller tags could record fine-scale motion, magnetic exposure, depth, and temperature throughout the crossing. Improved ocean models could separate intentional headings from displacement by currents and eddies. Controlled magnetic experiments on migrating adults could test whether specific field changes trigger turns. Such tests must reproduce natural depth cycles because orientation may shift with pressure or light. Until then, larval distributions mark spawning areas more reliably than dramatic claims about unseen gatherings. You should imagine the destination as a broad, dynamic habitat, not a pinpoint hidden on a chart. The decisive discovery may be a sequence of ordinary cues integrated with remarkable persistence. It may also reveal different strategies among individuals, seasons, or the two Atlantic species. Evidence could overturn favored explanations, because magnetic sensitivity does not exclude other compasses. The best model will predict real turns before the recovered tags disclose them. It will explain departures from Europe, America, islands, and rivers without erasing their different distances. It will also survive years when currents, storms, and temperatures depart from average conditions. For now, the eel's route is verified in fragments and interpreted through cautious experiments. Before dawn, the traveler descends again, carrying an inherited journey into cold blue darkness. Its navigation remains a living calculation between body, ocean, and Earth's measurable but partly unread field.